Good morning, everybody. My name is Brad Baker. I'm with the Ontario Bat Network. As you may be aware, we're a bit of a fledgling organization. We stemmed from the second Ontario Bat meeting, which was put on by the Toronto Zoo in 2019, with our main goal to ensure that a third Ontario Bat meeting was able to occur. As I'm sure you're all painfully aware, COVID has caused quite a stir and made in-person meetings impossible for the past year. So we decided as a group to get some presenters together and ensure that we're still able to get current research and bat work out to the broader Ontario bat community who may be interested. Today's presenter is Dr. Brian Hickey. He is a lead education and research scientist at the River Institute. Dr. Hickey did his graduate training in biology at York University, where he studied red and hoary bats' responses to changes in environmental conditions. His current research efforts are directed at evaluating the response of fish and wildlife communities to habitat degradation, restoration effects, and the ecological effects of water level variation. I think we're all looking forward to hearing what you have to tell us this morning, Dr. Hickey. So without further ado, let's get on to the presentation.
All right, well, thank you for having me. I'll just share my PowerPoint slides here. And so I'm going to talk today about variation in mercury concentration in bats. And this is work that has involved a number of people. Not everyone is listed here, but two of the principal people working on this project now are Bayley Bedard and Sarah Greenwood. And much of what I'm going to be talking about is their work that they're currently undertaking. So I work at a River Institute, which is an unusual place to find a bat biologist for some people. In fact, one of the first things that happened when I joined the River Institute, as its name suggests, is an institution that focuses on rivers. We do research, we do education, and community outreach, and our main goal is to better understand how river ecosystems work. And so I got hired there a little over 20 years ago, shortly after receiving my PhD working on bat behavior. And so one of the first things my boss did, my new boss, is he had a heart-to-heart discussion with me and told me, you know, it's great that you love bats, but this is a River Institute. You're going to have to work on aquatic things. And 20 years later, you can see from this first slide how well that conversation turned out for him. And so my first project at the River Institute was to convince my new employers that bats were part of river ecosystems. And so an alternate title to this research, talk could be how I convinced the River Institute that bats were aquatic animals. And when I first arrived at the River Institute, there was all this talk about mercury, and the mercury values and different components of the ecosystem weren't adding up. And I said, only half jokingly, well, it's because you should be studying bats. And this got kind of just laughed off as a joke, but I wasn't joking, because bats eat insects, and as many of you know, they eat large quantities of insects. I learned that very quickly studying the foraging behavior of this species in the image here, the red bat, which eats over 50% of its body weight a night, and some studies have shown even closer to 100% of an animal's body weight, a bat's body weight, is consumed. So they're eating huge amounts of insects, and many of these insects grow up as larvae in water, and mercury accumulates in aquatic sediments. Mercury contamination is associated with aquatic food chains. That's why we have fish consumption advisories in many parts, probably most parts of North America. And so they eat these aquatic insects, these insects that, at least as larvae or nymphs, are aquatic. And so bats, I told my new employers, were part of the river ecosystem. They have diets that are just as aquatic as many fish. They live as long as 30 years, so they're exposed to mercury for really long periods of time, and they're easy to sample. We can sample them without killing them, because mercury accumulates in hair or fur, And so I didn't want to sacrifice a bunch of bats to study mercury. The fact that bats have fur made it really easy to do some non-destructive um, sampling. And so I thought I had a great case for, claiming bats are an important part of the river and they're important animals to study from a mercury perspective. And this was back more than 20 years ago when there were no mercury values for bats at all in Canada, and very few in North America or worldwide, period. And so I was really enthusiastic about this, but my new employers weren't. They explained to me in a slightly condescending way, well, you might not know this because you're a bat biologist, but there's this process called bioaccumulation, which tells us that we only get really large values, large concentrations of mercury when you move many links up a food chain. So river otters, humans that eat fish are at risk, but bats feed atop such a short food chain, you're wasting your time. So I did what I usually do when people don't like my ideas. I ignore them, and I immediately went out to a hibernation site during swarming season, and I quickly captured 4 different species, and I measured mercury in 4 different species and found each of those 4 species had at least some individuals that exceeded 10 parts per million in mercury. Now, I'm going to assume that a lot of people here aren't mercury people. So to put that in perspective, if a human female had that kind of level of mercury in her hair, she would be expected to give birth to children with measurable learning deficits. And so we've seen this in native communities that were exposed to mercury, for example, when land was flooded to create the James Bay hydroelectric projects in Quebec, and native populations there were exposed to contaminated fish as a result of the mercury contamination associated with the dam construction. So all 4 species had at least 10 parts per million mercury. And these included long-eared bats, little brown bats, and big brown bats. And these are mean values, but all 3 species had at least some individuals exceeding that 10 parts per million threshold. So immediately I was told, yeah, but your sample sizes were really small. It wasn't a great study. All of those criticisms was true. But the point is, I only measured 3 or 4 northern long-eared bats, and already I found one that exceeded this threshold. So my point was, well, if I sampled 100, I probably have lots of heavily contaminated northern long-eared bats. So I followed this up, this research up over the years, and because I work at a river institute, I had no budget for bat research, so this was accumulated really slowly. And within a couple of years, I was finding even higher values, 20 to 30 parts per million, so 2 to 3 times the level that would be of concern if these were in human samples. If this was in human hair, we'd be concerned about women with those kinds of concentrations giving birth to children with learning deficits at 10 ppm, as I've said, and this is 2-3 times higher. And so once again, I didn't get a lot of supportive responses. I think all the chemists I work with thought, well, that's what happens when you give the biologist the keys to the chemistry lab. He comes out with crazy things and crazy statements. So I gave my samples to this guy who's a real chemist and was a colleague back at the time, Jonathan Hill. And he said, yeah, my numbers weren't great. I had underestimated the values. He was finding 40 ppm and more. So this got me interested and eventually got my colleagues interested. So the next step of the project was to look at cross-Canada patterns, and we related cross-Canada patterns to mercury in bats. So if you look at atmospheric deposition of mercury, deposition is highest in these red areas. So in the coasts, and in Quebec and Ontario, we're kind of in the middle, and then the Western provinces, things are lower. And so I collaborated with John Shuttle at Carleton and the Canadian Wildlife Service, and Alex Poulin at Ottawa U, and we put a call to biologists who are doing field work across Canada, some of which I saw joining us today, and we got biologists from across Canada to send us samples of bats so we could increase the geographic distribution of our data points and to answer the question, is this something peculiar to bats from Cornwall? Because a bit of history about Cornwall. Cornwall is an IJC area of concern. And so we have contaminated sediments that is a legacy of industrial activities in the past, in Cornwall's past. And so we wanted to know, are these bats just an anomaly because we're in an area that was heavily industrialized, which is known for contaminated sediments? And so what we found, combining our data that we collected from collaborators across the country, these are the green bars here, and combining other studies from the northwestern US, what we found is if you rank these sites from left to right, west coast to east coast, you find the highest values on the east coast, work by Yates and also Little et al. And you find high levels on the west coast, and Quebec and Ontario are in the middle. So Quebec and Ontario, where my samples were from, are already showing that some individual bats are really heavily contaminated, well above the threshold at which you'd expect biological effects. And yet these samples aren't even nearly as highly contaminated as individuals from the two coasts where atmospheric deposition is high. Obviously, once mercury is in fur, it's not affecting sensitive organs. So the other thing we did is we looked at correlations between mercury in fur and target tissues. And for lack of time, I won't go into the fine details here. But the bottom line is when you correlate mercury in fur against other organs, you get correlations between fur concentrations and many of the sensitive target organs, so brain, kidney, liver, fur, etc. So mercury in fur means mercury in these other organs. Fur concentrations in fur are much higher than in kidney and liver and brain. So a summary of what we know so far, or what we knew at the end of this study, is that the values of 10 ppm or greater, it's not an unusual anomaly. It's not something that was a result of a biologist trying to do chemistry. Some of these earlier measurements were in the days where we didn't have auto analyzers where you just stick the fur in and the machine does everything. You had to actually do benchtop chemistry. So it wasn't just lack of experience in a chemistry lab. These numbers were repeated several different times by several different labs. We saw that the patterns of mercury parallel atmospheric deposition. So mercury is a global contaminant. It's carried on, it's carried along with the movement of water as part of the water cycle. It evaporates, it moves. And so mercury in those high deposition areas on the East Coast and on the West Coast may find themselves in rain clouds drifting across the continent, raining down on other places. So where precipitation has high mercury, so do bats. So that probably shouldn't be surprising. And we also demonstrated that mercury in fur correlates with mercury in brain, kidney, and liver. Although mercury in fur, the values are higher. So But what it doesn't explain is why are bats so heavily contaminated relative to where they are in the food web. And so if you rank organisms from according to trophic level or diet, whatever you want to call it here, where on the left you have insectivores and in the middle you have omnivores, and on the right you have carnivores, we typically get high mercury concentrations in carnivores and very low levels in insectivores and omnivores, unless we're at a unique site where a site is heavily contaminated. So these are parts per million here. If you add bats to this picture, bats are down here in terms of trophic level, low on the trophic scheme. But their mercury concentrations are right off the chart here. And so we still, it still doesn't make sense. So my colleagues, when I first arrived at the River Institute, the story I told you at the beginning of this talk, they thought, well, you're wasting your time looking at bats. You're not even going to find mercury in bats. They have such short food chains. Well, I very quickly showed them that that was wrong. So what we're doing now, and this is the work of the new collaborators I mentioned at the beginning, Sarah Greenwood, and who's done some of the statistical analysis, and Bayley Bedard, who's working on the molecular side of things. So what we've done now is we're taking things from the broad cross-Canada scale, and we're trying to look at a much finer scale within Eastern Ontario. And we're trying to do two things. Well, we're trying to do more than two things, but two of the most important things we're trying to do is we're trying to capture bats, radio track them to foraging areas, figure out exactly what they're eating so that we can measure mercury concentrations in prey that they're actually eating, rather than just go out and randomly sample insects. Because there's several things here. So if we compare big brown bats and little brown bats, big brown bats are much more contaminated on average than little brown bats. And one of the challenges interpreting this data, though, is big brown bats tend to be associated with some of the urban sites, and the little brown bats we're catching tend to be associated with some of the more rural sites. So our big brown bats on the left here more contaminated because they're big brown bats, or are they more contaminated because they're Cornwall bats in many cases, and they're living close to industrially contaminated sediments. And so right now what we're trying to do is tease out the effect of big brown bats or species, big brown versus little brown, and location. Are they from an urban area? Are they from close to the St. Lawrence River? And that's some of the statistical analysis work that Sarah Greenwood undertook. So we took a very simple approach, a two-way analysis of variance, where we had bat species, just two species, big browns and little browns. We captured bats from 16 locations, and we measured mercury concentration as our response variable. We measured, the number now is higher, but more than 190 individuals. A lot of our locations, these are our locations here, a lot of them had really small sample sizes. So for our first round of analysis, and this is ongoing, so these conclusions are a little bit tentative. The first crack at the analysis that Sarah undertook was to take just the locations where we have both big brown and little brown bats represented. Our sample sizes for Somerstown and Williamstown for big brown bats are a little bit low. We're working on increasing that number. So this analysis is preliminary, but nonetheless interesting. So out of these 156 bats, the key part of this table here is the middle left table where it says analysis of variance. And under source, you've got location, species, and then an interaction effect between location and species. And so both location and species on their own are significant. Species is highly significant, and there seems to be a significant interaction between species. So big brown bats, if we just go back to this picture here, big brown bats are more heavily contaminated, whoops, because they're big brown bats, not simply because of the locations where they were captured, but location is important too, and there's also an interaction effect that's important. So I'll just get back here to where I was, and my slides seem to have frozen here. There we go. So, so both location and species matter, and there seems to be a significant interaction. So now we know that mercury varies across Canada in a pattern that matches atmospheric deposition. We know that both species and location matters, but we still don't know why mercury is so high in bats relative to where it should be based on their trophic position. There's been very little work on effects of mercury. If these bats were humans, we'd be really worried about the ability of these humans to function normally, learn properly, give birth to children who can learn properly and develop properly. And that threshold, as I said, is 10 parts per million, and we're catching bats regularly that are 20, 30, and 40 parts per million. So 2 to 4 times higher than that threshold at which neurological effects have been demonstrated. There is a little bit of work showing effects on mercury contamination and immune system function in bats. There's not a lot of work that's been done, but the little bit that has been done has shown that mercury can affect immune system function. I've been wondering for a number of years whether all of this, I'm sure most of the people in this audience will be more than familiar with white nose syndrome. You know, I'm wondering if maybe this devastating fungus is partly due to compromised immune systems because bats are exposed to such high levels of contaminants. That's complete speculation, but would be an interesting area for future research. We also don't know why big brown bats are so much more contaminated than little brown bats. Little brown bats, and anybody who works on diet of little brown bats, I'd be very interested in discussing this in more detail. I always think of little brown bats as being, if anything, more aquatic in their feeding habits than big brown bats. Big brown bats are common in cities, but little brown bats, if you want to go catch little brown bats, in my experience, the best place to go is over a pond, over a stream. And if you look at diet, data that's been published using molecular approaches to use kind of a DNA barcoding approach to figure out what bats have been eating. Little brown bats tend to have very aquatic diets. They eat a lot of aquatic insects. So I, going into this, thought little brown bats are going to be the more contaminated species, but it actually turns out to be big brown bats, and that doesn't make a lot of sense either, based on what we know about the diets of big browns compared to little browns. So that's why the radio telemetry component I talked about earlier is so important. What we're trying to do is track both of these species, figure out where they're actually feeding, and so that we can measure mercury concentrations in the same taxa of insects that we know these bats are eating. That's a really challenging process, and it's going very, very slowly. But we're giving it a try. The other interesting thing is if we look at locations where we have a reasonable amount of data, and this is some of Bayley Bedard's data. So these sites, if you look at the x-axis and we move from left to right, the sites that are along the St. Lawrence River are ranked from upstream to downstream. So Glen Walter and Somerstown are downstream of Cornwall. Morrisburg is upstream of Cornwall, and Martintown and Williamstown, the two sites with the lowest mercury concentrations, these are in a different watershed. These are not well, same watershed, different water body. They are on the Raisin River, which is a tributary of the St. Lawrence River. So they're not on the shore of the St. Lawrence River. So Cornwall is the place where all these contaminated sediments are. And if we look at the St. Lawrence River sites for now, forget about Martintown and Williamstown. Cornwall is the least contaminated. Bats from Cornwall, that is, are the least contaminated, and it's actually downstream Glen Walter and Somerstown and upstream Morrisburg that are most contaminated. And so if bats are contaminated because of insects feeding in contaminated sediments, you would expect the reverse pattern. So that doesn't make sense yet either. Interestingly, we often get the same pattern when we look at fish mercury. Although I've managed to convince my bosses to let me do bat research, I still do fish research occasionally to keep them all happy. And when I look at fish mercury, I see this same pattern. Martintown and Williamstown are interesting. Mercury is associated often with brown, high dissolved organic carbon waters, and the St. Lawrence River is very low in dissolved organic carbon. The Raisin River, where Williamstown and Martintown are located, is very high DOC. And so I actually expected that the Martintown and the Williamstown bats would be some of the most contaminated just because of the high DOC. So that doesn't make any sense. So it seems as though every time, if we look at non-bat literature, everything we think we know about mercury, bats seem to just tell us that this pattern doesn't make any sense. So there's something different about bats, and I'm sure all of you in this group know that there's something different about bats. They're way more cool than most animals, and that's why many of us study bats. I'm not biased at all. So now what we're trying to do is look into more detail at mercury concentrations in their prey. As I said, we're trying to do the radiotelemetry I referred to earlier. And another really interesting aspect, and this is the work that Bayley Bedard's doing for her master's degree, she's looking at molecular responses. And so I know very little about molecular biology. So if there are any molecular biologists in the audience, you may be cringing here at my explanations. But metals, when we're exposed to metals, when animals, including bats, are exposed to metals like mercury or other heavy metals, certain signaling cascades at the molecular level are activated, and they end up, the net result is the production of molecules that sequester mercury and either take it out of circulation or move it somewhere where it's not harmful or detoxify it. And so there are methylethionines, metal regulatory transcription factors called MTFs, and there are a number of different MTFs. Bailey's been looking at two of them, MTF1 and MTF2. And so what we're doing is we're looking at these MTFs, and what happens is when mercury enters a cell, it binds to one of these MTFs. It then enters the nucleus, and it binds to a promoter region in the of a gene, and it induces transcription of these molecules that help take mercury out of the solution. So these molecules bind with mercury and then either move those mercury molecules out of circulation or initiate other reactions that detoxify the metals. So what Bayley's been doing is looking at the numbers of gene loci and the number of these MRE promoter regions. And so for non-molecular biologists, which is probably most of the people listening here, what this means is the higher the number, the higher number of gene loci and the higher number of these promoter regions, the more of these detoxification or sequestering molecules the bats or the animals can make. So humans have really high numbers, 9 gene loci and 25 MREs. If we look at bats, little brown bats have 2 gene loci and only one of these promoter regions. Big browns, Aptesicus here, have 5 and 8. So it seems as though big brown bats, the species that are exposed to higher concentrations of mercury, have a greater ability to detoxify metals, perhaps including mercury. But once again, it still doesn't answer the question, why are we getting these values that regularly exceed 10 ppm on a species with such a short food chain that's at the top of such a short food chain? And why is the species that's usually considered the more aquatic forager, little brown bats, why are they more contaminated than big brown bats? So that is still a mystery. So, and how do they cope with these high levels? That's another question that I haven't even begun to figure out. So one hypothesis could be that they feed on aquatic prey, and they always have fed on aquatic prey. And even before humans started contaminating sediments, wetlands, probably we all here in this audience realize that wetlands are natural filters. They remove contaminants and they sequester them in the sediment. So wetlands, places where bats feed, have always been sources of mercury. And that's one of their ecological functions, to sequester contaminants before the water flows out into the main water body, which in my case is the St. Lawrence River. So maybe bats evolved more efficient ways of dealing with mercury because they've always been exposed to mercury, and maybe values of mercury that would be of concern to a human are not of concern to bats. So one of the things I'm looking at now, I have another student working on a project, and what he's doing is he's doing a meta-analysis of mercury data, and he's looking at ratios of fur to brain, fur to liver, etc., ratios in different species of mammals, to ask the question, is there something different? Maybe bats are much better at getting the fur out of the brain, out of the liver, out of the kidney, and into the fur. And maybe that's why we're getting these really high levels in fur, because bats have just evolved a better way of moving mercury out of sensitive tissues. So Bayley's working on the molecular mechanisms, and another student is doing this meta-analysis to try to give us some clues to whether maybe that's why bats are so different. So the next steps, Bayley's, I just gave you a snapshot of Bayley's work because I know so little about molecular biology, but she's looking at several detoxification mechanisms or pathways, global DNA methylation rate. She's looking at mitochondrial DNA expression, as well as some of these metallethionines and metal transcription factors that I've talked about. So that's what I've been up to, and I'm going to stop sharing my screen now. Happy to address any questions. If anyone's interested in developing some collaborations. That's how the River Institute survives, on collaboration. We're a small institution, and one of the great things about one of the few upsides about COVID is I usually can't get away to meetings like this when they're in Toronto, and this is an opportunity for me to just tell you what I've been up to, because I'm at a River Institute. I kind of don't meet regularly with all the other bat people in Ontario. So really happy to have an opportunity to tell you what I'm up to, and looking forward to hearing about some of your work in upcoming sessions. So thanks a lot. Happy to answer any questions.
I think I speak for us all when I say thank you, Brian, Dr. Hickey. It was a great presentation, and we really appreciate you taking the time to do this. There's a couple of questions in the, so Derek's question was, comment was, nice work, Brian. Do you expect mercury accumulation increases with age in bats? I understand that it's challenging because the difficulty to age individual bats in the wild, but what might be your prediction? I think FVU, or big brown bat, are expected to be less long-lived than little brown bat, but this is from little data. I'm afraid that I need to drop off the call, and that's where he ducked out. So the question is, do you think that age changes mercury accumulation within those bats?
Yeah, so one of the things we're doing now is the analysis of Sarah's that I presented. Bailey's now taking that the next step, and she's trying to add more factors into the model, and one of them is age. And in fact, older bats have higher mercury concentration. So I don't need to speculate on that. We actually have that data, and that's going to be part of the more fully fleshed out work that Bailey's in the process of putting together. So yeah, older bats, adult bats are more contaminated than young of the year bats. Now, we don't have anything where we compared 5-year-old bats to 10-year-old bats, because that requires banding and recapturing, and our sample size of those kind of fortuitous situations is if not zero, very close to zero. But that's one of these things over time I'm hoping to build up. So we do know that age is important, and once again, you'd think the longer lived species, little brown bats, they'd be exposed to mercury for longer periods. You'd expect them to have the higher detoxification rates, or the more well-developed detoxification mechanisms, but it seems like it's the other way around. So yeah, so once again, bats do everything differently.
And if Philip Olbaum would like to turn on his mic and camera, he could ask his question.
Hi, Brian. Great talk. I was just wondering if you're able to account for seasonal molting in any way, and if growth of new hair might be related to the diet, like during the period of hair growth, with the levels of mercury you're seeing.
Yeah, so that's a great question, and obviously hair is part of a growth cycle, and so we haven't looked at that in any detail, but that is something we've thought about. Now and I think now we're getting to the point where we probably have data now where we can do that, because a lot of times bats are brought to me because they woke up in the middle of winter and have been killed in an encounter with homeowners. And then we have bats in the summer that we capture and release. One of the challenging factors is any work we've done that's not with fur. So when we looked at liver and brain, all of these were bats that met their demise due to crazy homeowners, or frightened homeowners, or house cats. So because of that, it's not a very tightly controlled data set where I got 10 from this site at this time of year and 10 from this. So I didn't sacrifice any bats. All of the bats that we did tissue work on were just brought to me in that haphazard way because of unfortunate encounters with homeowners. But on the fur side, I think we have enough data now that we can probably look at seasonal trends. The other thing Bailey's looking at is trends over time, like over a span of a decade. That's turning out to be challenging because most of our samples are from the last few years and we don't have a lot of really old data. So those are things that we can't do yet, but I think we'll be able to look at soon. And it's definitely important, the hair growth cycle.
That answers your question, Philip?
Yeah, great. Thank you.
Perfect. Then we'll go to Ross Baker's question.
Hi there, guys. Thanks for inviting us.
A special hello to Melissa. You mentioned there had been limited studies elsewhere in the world on mercury levels in fur. I just wondered if any of those had shown similar levels of mercury to what you'd encountered in Canada.
Yeah, so since that comment was referring to the state of affairs when I began this work, now mercury research in bats has taken off in Canada as well as elsewhere, and the results from other places seem to be showing that bats in other places have elevated mercury concentrations as well. Some of the work is specifically at contaminated sites, and so it's not surprising. But more and more, we're finding mercury is elevated in a variety of different species. And there's a really interesting paper done recently, and I can't remember if it was Belize or it was either Central or South America, where they collected bat species, a variety of bat species that span a much broader range of diets than what we have in Ontario, which of course is where we only have insect-eating bats. And so they looked at insect eaters, fruit eaters, and even fishing bats. And fishing bats, not surprisingly, were really, really high because they're eating fish. So yeah, bats in general, not just bats in Ontario, not just bats in Canada, seem to be much higher in mercury concentration than what you'd expect, especially the insect eaters, which you expect to be low because the food chains are very short.
So that answers your question, Ross?
Yes, it does. Thanks. Can we ask
Another one as well. I think Lynn
believe so.
wanted to ask a question.
No.
No? Oh, she's okay.
It was answered before.
Right. Okay. Thanks
Sounds
a lot.
good. Thank you. So I don't see any other questions. I had a quick question about, you were talking about tolerance level of bats to mercury and then whether or not that might affect their immune system. And I was just wondering if you knew of or have seen any research done in Europe. As I understood it, the white nose syndrome came from Europe. And then I was wondering if we see similar trends in mercury deposition in bats in Europe and whether or not there's any evidence that it may affect their ability to fight it there.
I not that I have seen. Now, it's certainly possible that some of that work has been done, but I'm not aware of anything specifically connecting mercury to immune system function other than one paper. And off the top of my head, I can't remember the geographic location of that work. So yeah, so there's definitely not a lot of work on that subject, and that's work that if I stumble upon a collaborator that works on immune systems, I'd be really interested in doing something along those lines because I think it could be interesting.
Sounds good. We then have Adam Gratoli, pardon my mispronunciation if I did. He's just got a comment more than a question related to age. So I'll give him the floor for a minute here.
Hi there. You nailed my name, so that's great. Ryan, great talk. That was awesome. So I'm actually to speak to Derek's question in terms of age-related trends in Mercury. I'm actually doing my master's on that right now using a Mercury capture study. So we have first samples taken at multiple points over a bat's lifetime. And so far results seem to be showing that adults actually have decreasing, adult females, Myotis lucifugus, actually have decreasing levels of mercury in their fur. So it seems like there seems to be a sharp peak right from juvenile to adulthood, because we do seem to see that adults have higher levels than juveniles. And then once that peak occurs, there seems to be a slow decline in the amount of mercury in their fur. But these are just some results. My thesis will be coming out in a couple months if you want to take a look at that, or I'd be also happy to chat with you at some point if you're interested.
Okay, I'd be interested in both of those options. Yeah, so you can definitely get in touch with me. And where is your field work? Where are your samples from?
Newfoundland.
Okay, Newfoundland. Cool. Wow. After COVID, I'd love to have an excuse to go and visit Newfoundland. I've been there once. It's such a great province. But in the meantime, we can use Zoom or something similar. Yeah, no, really be interested in pursuing that with you, Adam.
Yeah, great. Yeah, I'll send you an email there then.
Okay. So that's so it sounds like you saw similar results to what we're seeing, that if you compare juveniles to adults, you get an increase. But the stuff you've added is really great, because being able to then see what happens after that adult burst, does it stay high, does it tail off? That's really interesting. I'll have to think about some mechanisms whereby that might happen.
So
yeah, for sure. I'd love to brainstorm with you, because I've been racking my head about that as well. So,
cool. This has sparked a few questions, if I might. Ken is asking if there is mercury transferred through breast milk, or if anyone has documented mercury transfer through breast milk.
That has been documented, yes. Mercury transferred through breast milk, I think. I'm remembering back to some pretty old work. Is that, would you agree, Adam?
Yeah, so reproductive mechanisms, there seems to be quite a bit of transfer, both from the milk and then also from the mother to the fetus.
So you might actually expect differences in that post-adult peak decline in males and females. Do you have enough to make that separation, Adam?
No, unfortunately we don't, and that's kind of one of the speculations I've been making as to whether we would see the same pattern in males as females. Okay. But we just have females, so.
All right, well that's okay, cool. So one of the things that, one of the other aspects of my research that I didn't talk about is most of my work in the last few years has actually been deploying bat houses, trying to just improve habitat so bats can stay happy and healthy and arrive at hibernation sites with a better chance of fighting off white nose syndrome. And I've got 4 or 5 bat house colonies that are quite large, given the context of this overall decline, with 50 to 100 or more bats. And so I've now got bat house colonies where I can potentially do those kinds of mark recapture long-term studies on known individuals. So maybe over time I'll be able to get some data that might address that question. Now, most of these will be females because they're maternity colonies. So the hard part will be getting males.
Excellent. So there also is a question here: are there emails that folks can reach you at? And I think maybe for just privacy purposes, it might be best if you guys are on the Facebook group for the Ontario Bat Network, that we carry on conversations related to contact information there, rather than posting this to YouTube for everybody. So that might be the
That's
best.
fine. And for those who are on now, if you Google River Institute or St. Lawrence River Institute, all of our emails are the same. It's at riverinstitute.ca. If you can't remember my name and can't remember B. Hickey, it's Bats at River Institute gets you to me as well. So yeah, so for those who are listening now or even on YouTube, I'm not too worried about people spamming me about bats, and then people who want to don't want their emails out, just email me. I don't mind giving that out on YouTube, and then you can and then I'll contact you.
Looks like we've got one more question here. Have you been in touch with John Shadlett at ECC?
Yeah, actually, he's the reason why I had money to analyze samples for the Cross Canada paper, and he's also part of Bayley Bedard's supervisory committee. So yeah, John Shadlett works on bats because of me. I went to him, and I don't think he had worked on anything bigger than an insect. He works really low down on the trophic level. And I went and said, I've got all these samples. I've got these students raring to go, but I don't have a mercury analyzer. And so, yeah, he kindly agreed to collaborate with me on that paper, and he's still collaborating on this work. So, yeah. And if anyone, John Shadlett is a great guy to collaborate. He's super, super nice to collaborate with. No disputes over turf or territoriality or egos with him. Those are the kinds of people I like to collaborate with.
Excellent. Perfect. Well, I'm not seeing any more questions here, so I think we can probably wrap it up for the day. And thank you again, Dr. Hickey, for coming out and presenting today. And yeah, I encourage everyone to carry on the conversation back on the Facebook page for Ontario Bat Network or via email. And we'll look forward to keeping in touch and getting more information out to everybody. Thank you very much, Brian.
All right. Thanks for having me, and thanks for listening, everyone.